Welcome to the webinar, everybody. This afternoon, I'll be presenting the example of fragment-based drug design carried out by the team at DomainX. And I'd like to start by telling you, with one slide, just telling you a little bit about DomainX. So we are located about 10 miles southeast of Cambridge city centre. We're currently 50 to 60 scientists. And we have protein scientists, assay biologists, and medicinal chemists all working together on drug research projects. This is our building here on the Science Research Park. And we're a dedicated CRO. So for the last 3 years, we've had no ongoing internal research projects. All of our work has been supporting client programs. And our clients range from big pharma through to academic institutions, but the majority of our work we do for biotechnology companies around the world, mostly in the UK, USA, Europe and Australia. And as a result of that work, a number of compounds have gone into clinical trials. Other compounds are currently in the preclinical development pipeline. And because of our contributions to those projects, we've been cited as inventors on more than 60 projects. The program I'm describing today was aimed at a group of inflammatory diseases known collectively as interferonopathies. This is a class of autoimmune diseases which are caused by upregulation of interferon signaling, and that leads to inappropriate activation of what's called interferon-stimulated genes, many of which are pro-inflammatory mediators. And major examples of these so-called interferonopathies are systemic lupus erythematosus, Sjögren's syndrome, and scleroderma. Just zooming in on lupus in particular as an example, there are about 500,000 cases of lupus in the G8 countries. And in fact, in China, there are more patients with lupus than with perhaps the better known disease, rheumatoid arthritis. Here is a photograph of a young person who unfortunately has lupus. As you can see, it causes these extremely painful and unsightly skin lesions. But even more tragically, it also leads to damage of internal organs, such as the kidney. Because of this, unlike some other immune diseases, it can be fatal, and a significant number of lupus patients, in fact, die of renal failure. Lupus can be treated with steroids and with methotrexate. And in fact, a drug that's very much in the press at the moment, hydroxychloroquine, is also used to treat lupus, but its precise mechanism of action, as per its muted action in COVID-19 treatment, is in fact unknown. In common with other immune diseases, there is a biologic treatment, a BAFF inhibitor called belimumab, but this isn't particularly effective and it's actually very expensive. And therefore, there is a very high unmet medical need for specific and effective treatments for interferonopathies. And that's why we were attracted to this particular disease class. As I said at the outset, our targets are IKK-E or IKK-epsilon and TBK1. And these kinases play an important role in the regulation of the immune response. You'll all know that the immune response has evolved to protect us from infectious diseases caused by bacteria and viruses. And essentially the way it works is that on immune cells, on the surface, there are toll-like receptors, and in the cytosol of immune cells, there are nucleic acid receptors. And these detect the products arising from the infections by these organisms and use those, and when they receive those signals, they stimulate the immune response through signaling cascades, to activation of transcription factors, to the stimulation of gene synthesis, of genes and then the synthesis of proteins that mediate the inflammatory response. In these signaling pathways, our targets are nodal enzymes. They're involved in both toll-like receptor signaling and nucleic acid receptor signaling. And so it seemed to us these were really good targets to inhibit in order to try and block this system in disease. In the disease, of course, unwanted pro-inflammatory signals turn on the pathways, and that leads to excessive stimulation of these pro-inflammatory gene syntheses. And they themselves are pro-inflammatory mediators, so we get into some kind of vicious circle where the system winds itself up. And our rationale was that if you could inhibit these enzymes, then gradually the system would turn itself down as the pro-inflammatory signals got weaker and weaker. So we wanted to tackle these targets, and we chose to use an FBDD approach. This was really because we wanted to firstly demonstrate the value of the approach, and secondly, because it had proved hard for other groups to find highly selective and bioavailable inhibitors using other hit ID methods such as high throughput screening. Now, I'm only going to talk about fragment-based drug design by implication. If you want to know the theory, I would commend this paper or review to you. But in essence, at Domain X, we start out by making the protein. Sometimes we have to use proprietary technologies to do that. We use that protein to develop assays based on biophysics or biochemistry, screen our fragment libraries, get structural biological information on how the fragments bind, and then use that to drive the drug design cycle. In this particular case, we screened the fragments using a high concentration biochemical assay against the IKK epsilon enzyme. And we were also informed by a crystal structure which we had uniquely obtained. It's still the only known crystal structure of IKK epsilon, which we used our domain approach to obtain. And then we optimized the compounds by molecular modeling and structure-based drug design using our cascade of enzyme and cellular assays. So this is the hit fragment I'm going to talk about today. I'm going to use this sort of top trumps representation so you can see some information about the compound. This is its potency against one of the targets as an illustration of the biochemical activity. This is the molecular weight. And these are two efficiency metrics that we use in order to quantify how good are we getting at finding a really nice compound. And this measure here, called ligand efficiency, tells us how well the compound binds to the target. And the value of 0.3 is considered very good. And the closer that we can get to 1, the better the compound is. We weren't able to crystallize this compound with the target, but we were able to dock it into the APO enzyme crystal structure that we had. And you can see that here. And you can see that it binds quite snugly into this groove. And I hope you'll be able to see that there are two opportunities to expand this compound, one in the northern part here, around this nitrogen, and the other one in this part here, which is around this position here. So we made analogues in both of these areas, starting with the northern part of the compound. You put a more bulky substituent here, and you can see you get a 10-fold improvement in potency, which is very exciting. And there's been no reduction in the efficiency of the compound binding. So that was a very nice start for us. We then said, well, what happens if we further substitute here? And we tried a range of things there. We tried directly adding aromatic rings. We tried adding substituents that would link through carbon or oxygen or nitrogen atoms. But the thing which really worked best for us was to add either an aryl or a heteroaryl ring with an amino linker. And that gave us a further 10-fold boost in potency. So we're now getting into the sub-micromolar range here. And this was very encouraging. However, we were slightly concerned by this drop-off in ligand efficiency. And one of the possible reasons for that is that, as you can see in this structure here, there is now a clash between these two hydrogen atoms in the enforced planar conformation of these two rings here. So we thought if we could remove one of these hydrogen atoms, then maybe we could get an improvement in potency. And what we found is if you replace this carbon here with a nitrogen to make this compound, you get another significant improvement in potency. And so with this scaffold here, you can see we've removed this clash, and we now have a 50 nanomolar compound. So far, so good. However, if you're familiar with kinase research, you might have think, oh, I've got a bit of deja vu here. I've seen this kind of scaffold before, this so-called aminopyrimidine scaffold, and you'd be right. And this is a really interesting example of what I think of as convergent evolution. So in a biologist, convergent evolution is when two organisms have evolved to be very similar, as a result of adapting to a similar environment. So here's an example of a bat and a bird. They're actually biologically unrelated, but they both evolved to fly in quite a similar way as a result of wanting to capitalize on the benefits of being able to fly. In this particular case, our environment is an ATP binding site that's largely conserved in kinases. And although nobody else, to my knowledge, has ever started with this starting point, it's quite interesting that everything we tried to do to develop this compound failed to give us good compounds, except when we converted it into an aminopyrimidine scaffold. So I think what this is teaching us is that natural selection, if you like, takes us towards this kind of motif, which has previously been discovered by other people. For example, in the marketed drug imatinib, you can see there's a very common, very similar scaffold here. Nevertheless, our fragment-based approach has given us this cyano group, which is very distinctive, and that cyano group for this particular pair of kinases gives us exquisite potency, selectivity, and novelty, and actually, interestingly, relatively clear IP space. So had we not pursued the fragment approach, it's quite unlikely we would have found this particular substituent. Here's one of our better resolved crystal structures of a protein ligand complex, and I've angled this so you can see particularly well here, I hope, how the cyano group fits into this very, very narrow pocket in the kinase. And it's hard to conceive of any group other than a cyano group which could have done that and also made a hydrogen bond to lysine 38, which sits at the back of this pocket. So by doing this, we've been able to get a very nice interaction with the target. So through this fragment approach, we've been able to find a motif that gives us high potency and selectivity. We now have to address the problems of getting good solubility, metabolic stability, and permeability. And one of the ways medicinal chemists use to do that is to lower the log D of a compound by introducing polar atoms and substituents. And what we did was to vary the molecule in a number of places, but the thing I'm going to zoom in on today in the time I have available is what we did with this phenyl ring here. So here's another view of that same crystal structure, and there are three possible isomeric pyridines that you can make, where the nitrogen goes here, here, or here. We made all three of them. Interestingly, this one, nitrogen here, is quite potent, but much less selective than the carbon analog. The one where you put the nitrogen here is much less potent, but very gratifyingly, the one where you put the nitrogen here is slightly less potent than the carbon analog, but it does crucially maintain that very high selectivity, and even more importantly, gave us very much improved metabolic stability. So looking at in sort of stick drawing format, we've gone from a molecule like this to a molecule with this pyridine in place instead. And as the final part of the optimization, we looked at modifying the substituent in this position here. I won't tell you exactly what we put in the molecule to get to our candidate today, but by modification of this group, we were able to end up with DMXD011. You can see it's a potent inhibitor. You can see its log D has reduced enormously from the original potent compound, and the aqueous solubility is hugely improved. It's actually 3 orders of magnitude better than this compound here. And as a result of these changes, we've now got a very low clearance compound, a nice metabolically stable compound. And this was certainly suitable for us to now go ahead and do some proof of concept studies. Some of those were done in animal models, but the one I'm going to show you today was done in an ex vivo human assay. So it's obviously crucially important to find out whether this might work in patients. And the way we did that was by taking inflammatory cells, either from healthy people or from patients with these diseases, and then treating them with those cells with our compound and finding out whether we could block the immune mediator release. And here are those data. So there are different colors for different types of subjects. So there are healthy people, SLE patients, Sjögren's patients, and scleroderma patients shown here. And when you stimulate these cells, you get release of interferons, and you also get release of other mediators whose expression is controlled by interferon. So we get a very dramatic increase in the level of these these inflammatory mediators. But when we apply the domain X compound alongside that, you can see we almost completely blunt that response. So it's a very encouraging proof of concept. We got similar data when we looked at the interferon stimulated gene profiles. So in summary, what we were able to do was to use fragment screening to find a very efficient and selective motif. We were able to use structure-based drug design to elaborate that to a potent series of inhibitors. And then by optimization of the PK profile, we were able to find a compound that could be used to demonstrate proof of concept in blood taken from patients. And I'd like to thank the Domain X team for this work and for you for your attention during the webinar. And I'm now going to field some questions. And I've got a couple of questions so far. The first was, does the Sano group engage in reversible covalent binding? And the answer is no, it doesn't. We've been able to demonstrate that by showing the reversibility by doing some experiments where we dilute the enzymes. Another question was, how important was LE and LLE metrics for narrowing down hits? And the answer was that ligand deficiency, the LE metric, is very important in our view. Lipophilic ligand deficiency is less important until you get into the optimization stage, and then we start to use it, but we don't use it to select the initial fragment hits because most of our fragments are of low lipophilicity. The last question was what FBDD screening method was used, and the answer to that is that we used a high concentration biochemical assay. However, we do also use biophysical methods for domain X, and the one that we most favor is microscale thermophoresis because it is fast, uses very small amounts of protein, and gives us a very free choice of buffers to use. But we also use other techniques like surface plasmon resonance as well. And then the other question I've had is X-ray crystallography essential for FBDD? And the answer is no, but it helps. In this case, it was very helpful. We have done FBDD programs where we haven't had access to X-ray crystallography, and there it becomes a much more empirical process. And the fragment screening took us, I think, about 2 weeks.